This podcast is brought to you by the Albany Public Library main branch and the generosity of listeners like you. What is a podcast? God, Daddy, these people talk as much as you do. Razib Khan's Unsupervised Learning.
The full audio of this monologue has not been uploaded. To hear the rest of the monologue, please go to razib.substack.com. Again, that's razib.substack.com. Hey, everybody. Welcome to the Unsupervised Learning Podcast. It's just Razib, and today I'm going to be talking about pigmentation, the genetics of human pigmentation, basically complexion, right? And also hair color, eye color, and whatnot. I'll talk a little about various things like albinism, normal variation, the genetic basis of it, and then also the evolutionary significance or lack thereof. It's a very interesting question for a lot of people because pigmentation is really culturally and socially relevant, but also it illustrates some aspects and elements of genetic dynamics that are quite illustrative of how Mendelism works. So basically, one of the questions that I get from people quite often, and so I'll start out with this, the question that I get, how is it that this person is a full sibling of this other person and their hair and eye color are totally different? And it could be maybe they're African American, hair, eye, and skin color totally different. And so the assumption or the question that I often get is, does that mean that the person that's much lighter has much more European ancestry? Because the average African American has about 20% European ancestry, and there's obviously going to be some variation within across siblings. I think standard deviation will be like a couple of percent, something like that. And so the question is, is this segregating out the European and African ancestry, and is this why that's happening? Well, yes and no. The yes part of it is on the pigmentation genes, it is. But on the whole genome, it might not necessarily be. So if I'm going to set an expectation, probably it's more likely that the person within sibling cohorts among African Americans has more European ancestry, but that is not necessarily so. And the reason is ancestry is a measure of your overall genome's origin, right? So if you're 20% European and 80% African, West African, that means when you look at the segments along your genome, and the segments are the sum of the 3 billion base pairs you have distributed across your, all your chromosomes, right? 23 chromosomes or whatever, 22 autosomes and sex chromosomes, right? And so that is a total reflection of your heritage across your genome. And that varies a little bit among siblings, but it doesn't vary nearly as much as physical appearance. And what I'm going to say is actually applies to facial morphology as well. There are a subset of traits that are kind of in between quantitative polygenic traits and Mendelian traits, monogenic traits. So a monogenic trait is, for example, sickle cell anemia. So that occurs at a particular gene, and it is due to, carrying the mutated allele. You can look at that gene and its edited and its mutated state, and that will tell you the trait. And there's a high correlation. We would say a high penetrance. So if you have this state in the genome at this Mendelian trait, you generally have this outcome, this trait or disease. At the other end, you have a quantitative trait. So, for example, height is, depending on who you talk to, 60 to 80% heritable, 60 to 90% heritable. Most people say at the high end. So that means that within a population like the United States or Britain, where food isn't a major issue, the vast majority of variation in population of height is due to variation in genes. So if you have a sibling who's taller than the other sibling, that is due to their genes to a great extent. And we know this to some extent by looking at how related siblings are genetically, because there's variation in that, about 3% standard deviation. And the more related the siblings are, the full siblings, the more they're correlated in their physical characteristics. So in this way, they're reflecting the same pattern among twins, where monozygotic twins, identical twins, tend to exhibit a much tighter correlation on height than fraternal twins, who are very close to full siblings, because genetically they are, even though their correlations are somewhat higher because they went through the same uterine environment. So, that's how it is with height now, but it's not a Mendelian trait. It's heritable. It's not genetic. Like, it's due to genes, but heritable is just a statistical property. So, for example, the United States went through a severe famine, and wealthy people kept eating, and the poor stopped eating. In a generation, you would have a situation where the trait is far less genetically mediated, it's far less heritable, and the variation in height is much more due to environmental factors. So the remainder that's not heritable is due to environment. Now, environment could be exogenous, it could be just the environment out in the world, or it could be interactions between the genes. It could be random stochastic things, random things that are happening in development because there's noise in biological processes. Okay. So that's a quantitative trait like height. Intelligence is like that. Most of the behavioral traits are like that, whatnot, right? And they're due to many, many, many genes, thousands of genetic variants. And so when I say thousands of genetic variants, I don't necessarily mean genes, they're within genes. So a gene is an X size, it's, thousands, millions, whatever. I think millions is on the large side, but in any case, it's many, 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 tens, hundreds of thousands of base pairs, kilobases, megabases, right? And so a gene in a molecular sense starts at a promoter, and it reads off the promoter, and it causes, the production of mRNA, which is translated into proteins, et cetera, et cetera. In a classic Mendelian evolutionary sense, a gene is just a region of the genome that is correlated with a trait output. And so when you're looking at the variation of the trait, you're looking at the variation of the gene. They're not that far apart, but there's some differences between what they call the analytic gene and the molecular gene. But in any case, genes are big. There are a lot of base pairs. And when I'm talking about variants, usually, but not always, usually I'm talking about mutations at a particular base, a C going to a T, an A going to a G, these sorts of things. And what that does is these little mutations change the transcribed messenger RNA, and that's how it changes the protein. And it usually breaks the protein, it messes the protein up. Sometimes it causes a new function, and that results in a new trait that's selected, right? So that's the molecular mechanism of what's going on in these mutations. And for something like height that's distributed along a bell curve, it's due to lots and lots of small mutations all across the genome. And there aren't too many. I mean, the effect size of the variation to the variation in height at any given position is very, very small. It's not quite infinitesimal. The original models, would say, oh, infinite alleles, infinitesimal effects, all these things. That's a mathematical abstraction. The reality is, we now know there are 19,000 genes. We now know that there are 6 to 8 million, whatever, 6 million polymorphisms in the genome. So that's what we have to work with in terms of the empirical distributions for humans. And within those, there's X number of mutations. I know with intelligence, something like 1/3 of the genome can be used as a predictive variable for intelligence variation. And so that basically means it's not causal, it's not directly implicated in some neurogenesis pathway or whatever, but the genes are linked in a way because there's so many positions that affect intelligence that large portions of the genome are tagged along with those causal variables. But that's just to get at the fact that we're talking thousands of markers that are causing variation at these quantitative traits, in contrast to the Mendelian monogenic traits that I talked about earlier. Pigmentation is interesting because it's kind of in the middle. Okay. A lot of people have an intuition that pigmentation is like a quantitative trait. And technically it is, because it's due to the combined effect of, say hundreds of genes, hundreds of positions. That's a large number, but it's far, far less than the thousands, tens of thousands that you see in, say, height or intelligence, the classic quantitative traits. Um, the issue with pigmentation is there's also really large effect loci. That basically means that there are genes that when you see a change in that gene, it's telling you something. I think HMG2, going off memory here, I think that is affecting height, and that is the largest mutation in the normal range. Obviously, there's things like achondroplasia, dwarfism, whatnot, but we're talking the normal range. I think that might have an effect in the homozygote state of like 2 centimeters, which is actually it's not trivial, but that's the biggest, and it's not necessarily, I think, common. So mostly you have to predict genetic height from these kind of complicated algorithms using all these positions. I mean, really, it's like polygenic score index with it's kind of like a regression model, right? Okay. With pigmentation, it's not like that insofar as there's huge effects. So some of the pigmentation genes explain as much as, say, 30 to 40% of the variation between two populations. So if you change the mutational state on a given gene, you will see a noticeable difference, not the whole difference, but a noticeable difference. And then you have a position like OCA2, uh, HERC2. These are two genes that are next to each other. Mutations in these genes cause the difference between blue eyes and brown eyes. Not exclusively. There's other genes and there's a variety of mutations, but there's a certain cluster of mutations at these genes that cause the blue eyes that you see in some humans, and it explains about 3/4 of the variation. Just this position, this particular locus. So it's not technically a Mendelian trait because it doesn't explain everything. In fact, the way I'm going to tell you is there's a combination of markers. It's called a haplotype. Basically, it's just like a little segment of the genome. If you see this combination of markers in Europeans, there is a 99 0.9% probability that the individual has blue eyes, right? And so what if you don't see those? Well, it turns out that a non-trivial minority of people that don't have this marker in Europe also have blue eyes. So what I'm trying to tell you here is you're not going to get a false negative if you find this set of markers. But in 25% of the cases, or 25% of the variation, there's other genetic positions that are also, other mutations in probably this locus that are affecting it. So that's just to get at the fact that it's overwhelmingly dominated by one combination of mutations, just like a Mendelian trait would be. But a Mendelian trait, we're talking like 99%, 100% of the variance is going to be at these little, subset of mutations within a gene. Whereas with eye color, it's 75%. So there's still some wiggle room in there. So sometimes they'll call this a quasi-Mendelian trait. It's not technically Mendelian, but it's close. Then you have something like skin color. So skin color is more due to more variance across many genes. Okay, now OCA2, HERC2, those two genes, they do actually affect skin color some, and they do affect hair color some. Pigmentation genes are numerous because the pigmentation pathway has a lot of inputs. There's some enzymes and some protein precursors, and if you can tweak with them in multiple ways in multiple parts of the pathway, and those parts of the pathway are controlled by genes, and so that's why you have hundreds of genes that can affect the pigmentation pathway. Okay. Hair color is kind of in between. It doesn't, from what I have seen in the literature, it doesn't seem as polygenic as skin, but it's not as clear cut as eye color. Often, if you just Google it, or if you go to Google Scholar, people will often say the KIT ligand. The KIT ligand gene causes blonde hair in Northern Europeans, but it also has effects on pigmentation, and I'm not sure if it would necessarily cause as blonde hair in non-Europeans. Like those of you on Clubhouse, that's actually an old picture of my son, but he carries one copy of the KIT ligand derived mutation, which is associated with blonde hair. His hair is noticeably lighter than his sister's. And my younger son is the same, actually. Does not look like that. That's after a solid summer in California. But so these are genes in pigmentation. You can see the effect visibly of some of these mutations. That's not true generally with height or intelligence. There's no particular gene that is going to have a big effect on a given individual. But with pigmentation, you see it. And so it's an interesting trait because you can memorize or just get familiar with kind of an alphabet soup of genes. So, for example, I've been tracking this. I've been tracking this trait for 18 years because in the year 2005, I started getting curious about inheritance of pigmentation in humans after I looked at some of the literature on mice coats. And there was a massive paper that came out in Science in 2005. Uh, and it looked at a gene in zebrafish, I believe. And it turned out that that gene, an ortholog, so that means the equivalent in humans, if you knock out that gene in the zebrafish, they get very pale. And if you knock out that gene in humans, they get very pale. And almost all Europeans are knocked out on that gene. The gene is called SLC24A5. So people would say this is the gene that makes people white and it's a mutation, et cetera, et cetera. That's actually it's quite the hyperbole. I'm actually a double knockout on that gene as well, and I am obviously not white skinned. So just like Europeans, I'm a double knockout. It's found the overwhelmingly dominant variant in the Middle East, and in South Asia it's at about like 50 to 60%, right? And so a lot of the variation in pigmentation that you see in South Asians is due to that gene. Same in Ethiopia and the Middle East. None of the variation in Europeans is due to that gene because there's almost no variation in Europeans. In some of the big samples, data sets of European genomics, you see that some individuals carry the ancestral variant of SLC24A5, which is overwhelmingly dominant in Africa and East Asia. And those individuals tend to have African ancestry in Spain, or they're in Finland. And why does that not surprise us? It's because Finns have about a 5% at least, maybe a little bit more, Asian ancestry. And so they carry the ancestral non-European variants of SLC24A5. But within Europe itself, it's at 100%. So this is what is called an ancestrally informative marker. In fact, the mutation at the specific base, I'm not going to name the RSID, but I think, yeah, I would recognize it. I've seen it so much. This is the second most ancestrally informative marker in the human genome, at least as of 10 years ago when I read that. I think particular. They might have found a different one. But that basically means if you find SLC24A5 in the derived mutated variant, which causes lighter skin, in other places, in Africa and East Asia, you should check for European ancestry. Or vice versa. If you find the ancestral variant in Europeans, you should check for non-European ancestry. And actually, before the whole genome analyses of Afrikaners from South Africa came back, I checked because this is a common marker to test for. So you can do it with PCR and other things, just when you're looking at particular SNPs, single nucleotide polymorphisms. And I saw that South African whites, Afrikaners that are not English, they had a couple of percent of the ancestral variant. Even at low sample sizes, I could see it. Okay. That was an immediate confirmation that genealogical researchers who claimed that, on the order of 5%, maybe a little less, but somewhere around there, of the ancestor of Afrikaners is African or Asian, that they were correct. Because if their ancestry is mostly Dutch and German, which it is, with some Huguenots from France, they should actually have none of the ancestral SLC24A5. And they did. So that immediately told me what the later publications which came out, I think Karina Schlischbach's lab, that showed the minority non-European ancestry in Afrikaner. So you can use it as a diagnostic. So these markers are have been extensively studied. And one reason they've been studied is they are associated to some extent with various forms of albinism and pathological mutations. So I've been talking mostly about normal mutation, but OCA2 is associated with a particular type of albinism Albinos, as you know, like they're hypopigmented, or they're hypopigmented. There's a variation in albinos actually that a lot of people don't know. Some albinos are not nearly as extreme, and they don't have white hair. They don't have the, like, I think like their eyes are kind of pink, but in any case, uh, many of these genes have been discovered through albinism and through mutations in, say, mice. So, when it comes to pigmentation, this is an issue where just looking at animal models, people have been able to find a lot of the genes because, they're all out there. Um, so, what has happened is, the gene names themselves. So I think like there's an agouti gene that's associated with guinea pigs. Again, like that has an effect in humans, associated with darker complexion in some populations. The SLC24A5 is a solute carrier. It's a transmembrane. It affects a transmembrane protein. There's another gene, SLC45A2, which is obviously out of the same class of genes, that is also associated with variation in pigmentation. If you break it, that also affects the melanin pathway. So I'm not going to go into the molecular mechanisms, but basically the melanin pathway has a precursor. I think it's like tyrosinase, uh, and that produces melanin from. So that's the enzyme that produces melanin from tyrosine, and so that's where it starts. And then it goes through a series of steps until it's a melanocyte, which is when you look at it under a microscope, the melanocytes are, there's several types. There's the pheomelanin production, which is the red, and that creates gingers. And everyone has that. If I bleach my hair, it shows reddish because of bleaching. Um, in any case, and then there's eumelanin, which is the brown one, and those look like within the skin, they look like dark granules, and people with darker skin have bigger ones and more of them. Right. This is what I recall when you looked at them, like under the microscope, and so that's the molecular, the end output of it. But all of these genes, what they're doing is they're getting into the pathway and they're upregulating or downregulating. So, a mutation at a gene, if there's one breakage, then it has half the protein and that has X effect. If you have two breakages, two loss of function, then that's really, really kind of putting a bottleneck in some position in the pathway. And that's how hypo or hypermutation, like down or up, occurs within the normal human pathway. So let's go back in terms of paleoanthropology to the past. There is the granddaddy of the of the pigmentation gene is melanocortin, MC1R. It's been extensively studied in mice. That's one of the reasons. And this is the gene that was known from the pre-genomic days, like in the 20th century. Geneticists studied the melanocortin loci, and they noticed it is associated. Some of them are associated with gingers. In terms of, if you have ginger hair, it looks like mutations in melanocortin can explain about 75% of that outcome. Right. So again, there's other genes, there's other mutations, there's different ways to get at the same output, but it's dominated in Europe by melanocortin. And when I say there's different ways to get at the same outcome, again, think of a metabolic pathway in a linear, because they're not all linear. Sometimes they have different things going on, whatever. But think of it linear as a step-by-step process, and there's inputs all along that process. So if you fiddle with the inputs, if you tune the inputs, you can modify the output in a variety of ways. So you can get the same complexion through different mutational architecture. And there's about there's about like a dozen big effect genes. So SLC24A5, the one that I mentioned that was discovered in zebrafish in 2005, is kind of the first big one in the genomic era. That explains about 30 to 35% of the pigmentation difference, that mutation itself, between people of African descent and people of European descent. So it doesn't explain all of it, but explains a lot of it. It's the largest effect locus. So if you think about a distribution and a power law, it's much more extreme than with say intelligence or height that I mentioned. So last, like going off memory, using genes that are known and annotated, I think Mark Shriver's lab has explained something like 50% of the variance, uh, just, using like 20, 30 genes. Okay. And then the rest of the variance he explained through ancestry. That means that there's residual genes that he hasn't explained. I think the newer work is getting closer to like 70, 80%. So I said that height is 80% heritable or whatever. It's 20% environmental. um I haven't actually looked at the heritability of pigmentation. It's quite heritable, obviously, but if there's gene interactions, developmental stochasticity in there, it might not be 100% heritable. There's going to be some noise in there, but it's not going to be systematic noise. Also, you should know that testosterone has an effect on the melanin pathway, and so men are darker than women. It's just like well known. In basically all societies where there's variation in pigmentation, you can tell that men are darker than women. It's like about 10%, I think, in the pigment. If you take steroids and other things that mess with your hormones, you can get darker. As we age, males and females, we both get darker, but women may get a little lighter during puberty as their estrogen testosterone balance shifts a little, and then as they hit menopause and later, their complexion and they get darker. And we see this really, really clearly in the hair, where young children can be very blonde, and then their hair is dark as an adult. Pigmentation is occurring over your lifespan. This actually occurs the same way among Australian Aborigines. There are some Aborigine groups where blondism is present among women and children, but very, very rarely among adult males. So I think you're seeing the same um, biomolecular pathway here, where testosterone among males is making them darker. And so, women can be lighter and children can be lighter, right? So there isn't that much work on the evolutionary origins of like what, say, the modern human looked like. I think in 2007 or whatnot, there was some work looking at the melanocortin 1R region, which is, it's a big. There's actually several melanocortin genes, but that's why there's one. And what they found is it looks like there was a selection event in the region of that genome, and based on the variation that they see in Europeans who have mutated and diverged, in particular, although other groups have too, from African population, they inferred that the selection event occurred like about 1.2 million years ago. What does that mean? So the hypothesis and why they were studying it is that they think that this is when our lineage Homo, or like on the border of pre-Homo, actually, lost its fur. I doubt we were ever as intensely furry as chimps. We were savanna apes, but if we had fur, if you have fur, your skin doesn't get exposed, right? And so some of these apes that you see, like say adult male chimps, have very dark faces. Again, the baby chimps can have pink faces, but the adult male chimps have dark faces. They get, if you shave them, they're still pale underneath. Sometimes they're not. It depends. A lot of them, they look kind of grayish, I think. But the point here is you don't need a lot of melanin production in your skin if your skin is covered by fur, because that's blocking the sunlight. But once you lose it, you you have exposed skin. And so what's the problem here? A lot of people think of skin cancer and that sort of stuff and sunburns. Like those are really, really annoying. But skin cancer is a late in for most people. I mean, okay, if you're a ginger at the equator, it's bad, but there are probably never gingers at the equator. So in any case, that's that's bad. But Nina Jablonski is a physical anthropologist who's done a lot of work on skin, and she has a model which to me is the most persuasive. And basically, many of women take folic acid to prevent neural tube defects, causes miscarriages, and basically, the process of the sunlight hitting your skin it destroys like the folate production pathways. And so then it could be that extremely light skinned women at latitudes where there's a lot of sunlight, we'll have many, many more miscarriages. So this is an evolutionary reproductive hit that's extremely strong. And so what would have happened after we lost our fur is we would develop, hyperpigmentation, compared to our ancestral state, which was, furred and paler, probably. Our faces, I'm assuming, were exposed and darker. If I'm going by our relatives, right? Chimpanzees, gorillas, whatnot. In any case, so we are this African population with darker skin. Neanderthals and Denisovans leave Africa about 700, 750, 800,000 years ago, maybe a million. So this is before STEM humans, anatomically modern humans, split from the Neandersovans, who themselves split later on. In any case, you have these dark-skinned human beings now. This is where we are, and they spread out of Africa, right? And today we see a huge range of complexions. People in East Asia, in Europe, and to some extent the Middle East, are fair-skinned relatively. People in Africa, in Melanesia, which is called Melanesia for a reason, very dark skin. Australian Aborigines tend to be dark skinned. The indigenous Negritos, they're called Negritos. Because they're small island people in Southeast Asia are dark skinned. Many Indians are very dark skinned. Some Indians are very light skinned. So there's a lot of variation within that population. And then you have situations like, in the New World, people tend to be kind of a coppery, like a light brown to a dark brown color, depending on where they are. So in Peru, the Peruvian highlands, people are darker. Whereas in the Amazon, they tend to be a bit lighter skinned. So there's still variation in the New World, even though the range is range is narrower, though, right? And this variation is due to mutation. So one model that you can have is that as people move northward, they don't need the dark skin anymore. And there's like the mutation rate at a given base is 10 to the negative 8. You have enough replications. Eventually, you're going to have a breakage. But if it doesn't matter because you're not going to have neural tube defects, then maybe the mutation will spread just through random genetic drift. Really, what will happen is some sort of mutational accumulation. And eventually, one of the mutations will actually drift out of low frequency and go up to fixation, right? So this is a model. And the model kind of works for red hair, but it doesn't work for all of the genes. And so what started happening in the 2000s between the aughts is you started using genome-wide association, looking at people with variable complexion and looking at parts of the genome that are giving off signals. Now, you need really, really large sample sizes to do this for quantitative traits and, kind of like sophisticated methods. Anyway, I don't want to get into that, but with pigmentation, you don't need sophisticated methods. So pigmentation immediately was figured out by the earliest genome-wide associations using relatively small sample sizes. There's a couple of reasons. One, the number of genes that control the variation is small, and they have a large effect. So this means that the effect size is large, and your sample sizes are not really underpowered compared to what you're trying to explore. Now, they didn't know this ahead of time. But they were lucky, so they started getting these like big, large effect genes. Also, there's a finite number of genes that affect this melanogenesis pathway among metazoans, among vertebrates. So it's just like the same genes over and over again. We kind of actually knew, even with candidate gene studies, where to look because we had results from mice. So a lot of these genes were actually discovered in variation in animals. So the KIT ligand gene, which is responsible for two things. One, blondism. It's about like 20% derived variant in Northern Europe. Both my sons have it, a copy of it. And so it's 20%. So most people are actually heterozygous like them. I think it's also associated, though, with testicular cancer. So a lot of these genes are associated with other side effects. And so that's important to keep in mind. That's called pleiotropy. It's when a gene has all these, it's involved in a lot of different things. It's complicated, right? Okay. So, we're discovering all of these genes about 10 years ago and trying to figure out how they can predict things. So it's a really big deal in forensics. You probably know about Hirisplex and all of these forensic predictors. About 10 years ago, there was an article about how, there was a murderer. They got his DNA and they predicted. They said The witnesses claimed that it was a Hispanic man. I believe this was in Louisiana and New Orleans or something. And it turned out that the individual quite clearly had fair hair and blue eyes. Now, that could still be a Hispanic. It turned out it was just a misidentification. And people were freaked out that you could forensically reconstruct an individual like that. Today, this is trivial. Today, we're forensically reconstructing ancient DNA and all sorts of people. And it's trivial. So with these SNP panels using training sets of modern individuals, you can get pretty good prediction. I already told you if you have one particular haplotype in OCA2 HERC2, you can do almost 99% prediction that someone has blue eyes. Now, the other eye colors like brown and green, they're a little bit more difficult, partly because there's more variance segregating and, there's still some work to be done. Although this, for intelligence, we have sample sizes of 3 million. We don't have that last I checked for pigmentation. If we did, I think this would be easily solved because it is actually a much more practical problem than in height or intelligence variation, right? So we have these like forensic predictors and we're using them. Now, one thing that people started noticing is they're not as good in non-Europeans. They're not totally bad. So SLC24A5 seems to have occurred, mutated in the Middle East somewhere a long, long time ago, far, far away. No, not far, far away. But it spread into Europe relatively recently, and I'm going to get to that story because we know a lot about the pigmentation history of Europe. But it also spread into Africa with back migration from the Middle East. It explains a lot of the variation of populations like Ethiopians, of their variation. But there's still other genes out there. So there had to be studies within Africa. There is a gene in places like the Sudan, which has a mutation. The gene starts with an M. It's really long, and I'm not going to say the whole gene, but I'll just say this is the hyperpigmentation gene. I think that'll be an easy way to remember it. And Sudanese and people like the Luo, which is Barack Obama's biological father's Luo, they're noticeably darker skinned than their Bantu neighbors, and obviously Egyptians to the north, but definitely even their Bantu African neighbors. And it's just noticeable. Well, the Sudanic zone is mostly savanna, and it's on the edge of the Sahara, and it has incredibly high rates of UV ultraviolet radiation. Thank you for listening. To hear the rest of the monologue, please go to razib.substack.com and subscribe.
This podcast for kids